Hey, and welcome back to Lanterna Biology. Today we're going to focus on the details of photosynthesis. So in the last video, please go and check that out if you haven't done so already, we spoke about metabolism again, and we focused on the relationship between cell respiration and photosynthesis, but we only covered sort of the broad strokes of photosynthesis itself. That is enough if you're a standard level student, but if you're taking HL, you definitely need more of the biochemical details of that metabolic process. And so today's video is going to give you just that. It's a bit nitty-gritty and a lot of details, but if you bear with me, I promise you we can get there. If you're an SL student, you might also benefit from the details, but you're definitely not obliged to know all of what we're going to discuss. So let's jump right in and let's go back to our light-dependent reactions, which is the first part of photosynthesis. So here we have the sunlight coming in and we're located in the thylakoids. Again, the thylakoids, those are sort of the pancakes, that was our analogy, the stacked pancakes. So we're in the chloroplast, right? And we have separate membrane-bound sacs, the thylakoids. And in those membranes, we have so-called photosystems. And they're actually, if we look at the process, there are two photosystems that are going to be important for us. Funnily enough, the first one is called photosystem 2, and the second one photosystem 1, because they were discovered actually in that order. What are photosystems? It's essentially just photopigments that are inserted into the thylakoid membrane. So chlorophyll, for instance, which is a photosynthetic pigment, is part of that. And so the light hits these photosystems, and as the light hits the photosynthetic pigments, which by the way are different for the two photosystems, that's why there are two. So they absorb different wavelengths of light. And as the light hits the photosynthetic pigment, we excite electrons that are in the photosystems. And so again, exciting an electron means bringing it up to a higher state of energy. And then that we can use in an electron transport chain. So what happens is that, again, photosystem 2 is actually the one where that happens first. And so our excited electron can then be passed down in electron transport chain, and we're using it in the carrier molecules that are also embedded in the membrane to, just as it also happens in cellular respiration, to create a proton motive force. What that means is we're taking H+ ions, so protons, hydrogen ions, we're taking them and we're translocating them, that's active transport, so we need the energy from that high energy electron. We're transferring them in order to create a gradient so that we have more H+ ions inside of the thylakoid than in the stroma of the chloroplast. We have a higher gradient there, and then later we can use that gradient of hydrogen ions. So we have a proton motive force. And as you hopefully remember from what we learned in cell respiration, that happens by using ATP synthase. So that is a transmembrane molecule that is actually an enzyme that catalyzes the phosphorylation of ADP, adenosine diphosphate, to adenosine triphosphate. So we're adding the phosphate group, creating ATP, our energy currency of the cell. And so that entire process is called photophosphorylation, because we're using light, photo, in order to phosphorylate the ADP into ATP. And now we're actually getting to our photosystem 1, because we pass down the energized electrons from photosystem 2 down the electron transport chain in order to carry out photophosphorylation. And now the de-energized electrons are in photosystem 1. Photosystem 1 has pigments that absorb light at a different wavelength. And so now we can re-energize the electrons using light energy again in order to carry out one further step. And this last step is a reduction reaction. We're using our electron in order to reduce NADP+, which is an electron carrier. And so in this reduction of NADP+, we're adding the electron to this molecule, so we get NADPH, because reduction is gain of electrons, and so also gain of hydrogen. So NADPH is what we get, that's our result. So that is a reduced electron carrier carrying this electron, which we'll use in a further step in the light independent reactions. Now we also need to replenish our electrons, right? Because we just send off that electron that started out in photosystem 2, was energized, used in photophosphorylation, re-energized in photosystem 1, but now it's gone because we gave it to the electron carrier. So what we need to do is we need to get electrons again for our photosystem 2, so the first one, because the photosystem 2 electrons were given to photosystem 1 and then off. And we get this in a process called photolysis. So you can kind of guess what that means from the word. It has photo and lysis in it. So photo, light, and lysis, to split something. And so photolysis is the splitting of water, the splitting of an H2O molecule by light energy. And so splitting water gives us H plus ions and oxygen, right? And so oxygen is given off as a byproduct, and this is why photosynthesis creates oxygen, right? It's just this little tiny step that has such huge implications, of course, for the environment, for the development of life on Earth. This is where the oxygen comes from splitting H2O water in photolysis. And we don't need this oxygen, right? It's given off. What we need is the hydrogen, the H+, for creating later on our proton motive force, our gradient of hydrogen atoms. And also we need the electron, right? Because we need the electron to put it into photosystem 2, and then later on it gets passed on to photosystem 1, and then on to the electron carrier, to the light-independent reactions, which is our next step. But just before we get there, a little mnemonic device for this entire part of the light-dependent reactions is a Z or Z, right? Because if you kind of imagine this, we put the electron, which is not energized, in photosystem 2, we bump it up, we energize it using light energy, then it gets de-energized in the electron transport chain. We're in photosystem 1 now, and it gets re-energized. So we have this kind of a pattern to this, right? And then once it has been re-energized in photosystem 1, we know that it's given off to the electron carrier. Following our light-dependent reactions, we get into the light-independent reactions, the second part of photosynthesis. They're sometimes also called the dark reactions, or you can just refer to them as the Calvin cycle, which is the biggest part of it. And so the light-independent reactions are taking place just in the stroma of the chloroplast, so not in the thylakoid or across the thylakoid membrane anymore, because in this part we don't need the membrane. And we're using, of course, what we just produced in the light-dependent reactions, but we're also taking in some other molecules, because again, think of what we want to do in the end. We want to produce an organic molecule. So here is where carbon fixation takes place, because we need, of course, carbon for that organic compound. The first step is the carboxylation of RuBP. So that sounds really weird. Let's kind of take that sentence apart. Carboxylation means we're adding carbon, right? We saw decarboxylation in cell respiration. Here, of course, we have the opposite. Because we want to fixate the carbon, we need to carboxylate. The Calvin cycle is, as the name suggests, a cycle. What does it mean if we have a cycle? It means some sort of molecule always stays in the cycle. And so we're looking at one particular moment where we add something to that molecule, then the cycle happens and we're modifying this molecule, and it ends up the same again so that the cycle can start over. And the molecule that we're looking at in this moment when we add something to the cycle is called ribulose bisphosphate, or in a short form, RuBP or RuBP. And so this is one of the most common molecules on Earth, actually, because all plants have it, and you know how many plants there are. And so RuBP is very central here. So we carboxylate RuBP, which is a 5-carbon compound. So that means we're adding carbon, that which we get, of course, from CO2, from the air, and we add that to RuBP. In order to do so, we need an enzyme to catalyze the reaction, and that enzyme is called RuBP carboxylase. ASE at the end, that means it's an enzyme. So carboxylase, of course, because it catalyzes the carboxylation. And the short form for this enzyme is Rubisco. So we're using the enzyme Rubisco, or RuBP carboxylase, to catalyze the carboxylation of RuBP. That is the 5-carbon compound that is in the cycle already. If you look at the numbers here, it's a bit complicated because this actually happens 3 times. So we have 3 RuBP, and for that entire thing, if we go through the cycle now, that gives us half a sugar. So we need to do the cycle twice to get one sugar, one glucose molecule, so one 6-carbon compound. And in each cycle of these two, we have 3 RuBP to start with. So it's a total of 6 RuBP for one glucose molecule. But let's stay in the cycle for now. So we have RuBP, we add CO2 to it, or just the C of the CO2 actually. So now we have a 6-carbon compound instead of a 5-carbon compound. However, that compound is unstable, and so it breaks down into 2 3-carbon compounds, which are called glycerate 3-phosphate, or just G3P, or just GP. So glycerate 3-phosphate, the 2 3-carbon compounds. And again, thinking of the numbers, we have 6 of those, right? We start with 3 RuBP, but as they then get more carbon from CO2, they break down. So we have 6 GP, and they are now being reduced. That's our next important step, the reduction of GP, of glycerate 3-phosphate, using, of course, our NADPH, the reduced electron carrier, in a redox reaction. That means we're reducing G3P. The resulting compound is called triose phosphate. That means we're oxidizing NADPH into NADP+. So we also need 6 of those, right? We need 6 NADPH. And we also need 6 ATP because we need energy for this process, and the hydrolysis of ATP to ADP gives us that. And now it is the triose phosphate that is actually our closest thing we now have to getting glucose. So this is what we're actually going to use. But because this is a cycle, we can't take all of it. We can't take all of the 6 and just then make glucose out of it, right? We could make 3 glucose molecules out of it, out of the 6, right? Glucose has 6 carbon molecules and a triose phosphate has 3. But we're actually just taking one. Just one of the 6 is taken to form glucose. The other 5 stay in there to regenerate RuBP. So that's why in the end we need to run the entire Kelvin cycle twice to get one 6-carbon glucose compound, because we just take one triose phosphate per cycle out of the cycle for glucose formation. So the remaining 5 triose phosphate molecules stay in the cycle, and we're actually expending more ATP now in order to regenerate RuBP and stocks of RuBP that we can keep from those triose phosphates. So now I know that was a lot of information at once, but I think we kind of got to the point of the details here. Again, the video before on photosynthesis is important to understand this, and we sort of go into more detail on the surroundings of it, the entire metabolic process, but here are now the details of the light-dependent reactions and photophosphorylation, and sort of that up and down of the electron being energized, that energy used and re-energized by light energy in our 2 photosystems, and then using the NADPH, the reduced electron carrier, and the ATP that we produced in the light-independent reactions, or the Kelvin cycle, and then looking at the 2 important molecules, RuBP and Rubisco, the enzyme, seeing how we can carboxylate that, and then also seeing how important it is to keep a lot of the material actually in the cycle, and we have to run the cycle twice just to get a single monomer, so a single glucose molecule. But then we have that, and so 2 important things. In the end, we get our organic compound, right? What happens to that then? Well, it goes into cell respiration. So these 2 processes, cell respiration and photosynthesis, are really linked. Of course, plants carry out cell respiration themselves, but also then once anyone, a rabbit or a horse, whatever, eats plants, right? Or you as a human when you eat spinach. You then take, of course, those organic compounds in that were created by the plant and then use them in cellular respiration inside of yourself, inside of the organism. And then also the oxygen that was created, if we remember that, the oxygen as a byproduct of photolysis in the light-dependent reactions, because we just want essentially the hydrogen and the electron from the H2O from water. But then we also get the oxygen, which is put into O2, so atmospheric oxygen, just as a byproduct. But that is actually really great because we need that in cell respiration. And if you don't remember where we need oxygen in cell respiration, go back to the video on cell respiration and watch that, because it's really these links and links between different topics or different processes here that kind of like solidify the knowledge in your brain, and then you can be sure that on the next test you will definitely remember that. So if in this moment you can't think of where you use oxygen in cell respiration, make sure you figure that out now. Go and watch the video on that one. And otherwise, I'll see you in the next video, and I hope this was really helpful to you.